Are you ready to science? What's up, everybody? Welcome to class 2. Are you as excited as I am? I don't know if you should be. Let me see. Let's make sure this is all running okay. Make sure I can see the chat and everything like that. All right. Hey, Ethan. Hey, Matt. What's up? How's it going, everybody? We're ready to work with some human cells again today. Matt, I think you asked last week if you could grow cells on all parts of the flask, and I actually tried, and I found out that you can't. So only the bottom part of the flask, it appears, cells can attach to. Let me see if I can find. Yeah. So I scraped this flask. Let me bring my microscope. I scraped this flask and I just flipped it over, and it doesn't look like any cells attached. But I actually didn't look at it under the microscope yet, so we can do that. Let's check it out. Because these flasks, you know, they're treated so that you can grow cells on them, and I don't know exactly what the process is of treating them. Yeah. So forgive me. But yeah, so I tried to grow it on other parts of the flask. The interesting thing is that like cells can grow on just glass. Obviously, glass is more expensive and you have to clean it and all this other stuff. So that's why everybody switched to plastic. But originally, everybody just used glass. You can just use any type of glass, really, and cells will grow and attach to it. And I've just used like baking glass and stuff like that before. So it's not something that has to be extremely specific. Well, actually, I stand corrected. It looks like the cells did attach to the top of the flask. That's funny. All right, let me bring up the video here. All right, so this is the top of the flask. When you look at it, you know, usually when I look and see if there's cells growing, I'll hold it up to the light like this and just look at it. And when I do that, it's hard to tell. But I guess when I just flip the flask over and put it on the microscope, you can see that there are cells growing. Yeah, so today we're going to go over changing the media, splitting and passaging the cells, and everything like that. So yes, you need to change the media. You can see here's, all right, let me switch back to me. Oh, don't turn down the temp on your cells. No. Ooh. Yeah, so don't turn down your temp on your cells, Jewel. Like, that doesn't work. So like with other cells, you can bring the temperature down and they'll grow slower. It doesn't work with human cells. You'll just kill them. So don't bring down the temperature on your cells. Generally, what you can do is just seed a new flask with very little cells, and then they'll grow slow. But don't turn down the temperature it won't slow down your cell growth. It'll just kill your cells generally, right? You have to think of these cells, human cells, like a human body, right? If you just put your body at a really cold temperature or a cooler temperature, you're just going to die, right? We have a very narrow width of temperatures that we can survive at as human beings. So don't bring your temperature down. Yeah, but anyway, so you can see the media. So this one is the scrape culture I did, and this one is the original culture. So you can see how the pH changes. Generally, when your media starts to get more yellowy or like orangey, that means you should change the media. Yeah. So see, one of them is pink, one of them is like orangey. Generally, when it's pink, it's okay. When it's orangey, not okay. So you should have received a heat mat, Robin, and the heat mat should have a temperature sensor on it. And the temperature sensor should allow you to keep your cells at the proper temperature if you, you know, and we talked last week about using towels and using other things, using styrofoam containers to keep your cells at the proper temperature. Ethan, could you overheat cells? Generally, no. I mean, obviously, if you bring your temperature up to like 42, 45 Celsius, you can overheat your cells and kill them. But if you're using a heat mat with a temperature sensor, it shouldn't be a problem. Right? As long as you're monitoring the temperature and you're keeping it at around 37 Celsius, you should be okay. It shouldn't be that big of a deal, right? But yeah, so we, so you probably, if you've received your cells yet, I know some people ordered late or not everybody's received their cells and everything yet, or some people's cells died. If your cells have died, email us, let us know. We'll reship you cells just for the cost of shipping. So just let us know. We'll just send you more cells, right? We want you to be able to do this class. We want you to be able to grow, learn to grow and engineer human cells. So if you're taking this class, if you signed up and paid, just contact us and we'll send you more cells for the cost of shipping. It's not a big deal. Just email Odin at the-odin.com. Don't contact me personally because like generally I'm not the one who takes care of that. And it'll just get lost. So email Odin at theodin.com or something, and we will, you know, we'll figure out how to get you more cells fast this week. If your kit hasn't gone out yet, if you just made an order recently or something like that, it should go out tomorrow, Monday. So you should have everything. Next week, David Ishii is going to be teaching the class. I'm going to be traveling in Ukraine, hanging out with Daria, Dan Siva, and filming for our biohacker documentary project. Yeah, so next week Dave is going to be teaching the class, but this week we're going to go over changing the media, splitting the cells, and all that good stuff. So you should right now, or when you get your cells, after a week, you should have a backup flask that's looking pretty good. Here's my backup flask that I started last week. Yeah. So here's what my backup flask looks like. The cells look pretty good. You know, they're growing, they're doing well. So when you scrape and do a backup flask, you know, it might take about a week, maybe a little bit more, but you should get cells that are looking nice and healthy. Look at those cute little human cells doing so well. Your backup flask, you don't really need to change the media on it until the color starts to get orangish yellow. Just keep it, leave it. Let the cells grow. It's just a backup flask. just in case other shit goes wrong. Oops. Can I bleep that out? I'm trying not to use profanity. That's like my new goal. So yeah, your bag of Flask, just keep it in your incubator. Don't worry about that. We'll let it go. Now, everybody should have gotten a bottle of trypsin. Should look something like this, probably. Trypsin versin or trypsin EDTA. These are what we're going to use to passage cells today. So what we need is we need some media. So get your media. You should have been sent a bottle of L15 media with 10% FBS and antibiotics. So we need that. And do I got some media? Yes, I got media. And we need the trypsin. So one important thing, so trypsin is a little enzyme that likes to cut up proteins. So our cells are attaching to the surface of flasks using proteins, right? So they just like stick out some proteins and like attach to the surface of flask. This is my performance art of me being a cell attached to a flask. I hope somebody took a picture of that. So the cells attached to the flasks, and we need to remove them from the flask. Now, we could use mechanical separation like we did last week, right? Where you just kind of like scrape off some of the cells and then move them to the new flask. Though generally that doesn't work as well and is not as, it doesn't help the cells proliferate and grow as fast as you would like them to. So instead, what we're going to do is we're going to use trypsin to cut up those proteins so that the cells just get off the flask, and then we can resuspend them in other flasks, right? So what you're going to need is some extra flasks. So generally with HEK cells, they're pretty proliferative. So like you can use a very small amount and it'll grow a real lot. So like one flask, you can split into 4 or 5 flasks if you want. You can even just use a very little amount if you want them to grow really slow. So if you're like traveling or doing something and you don't want your cells to overgrow and use up all the media and die, what you can do is just take a tiny bit of cells and put them in a new flask with HEK cells, and over time they'll just start growing out. But generally you want it to happen much faster, especially for experiments, right? We want everything to happen for this class within like a week, at least. So that's what we're going for. So generally, I imagine you probably have pretty confluent HEK flasks. So here is mine. Wait, let's see. Yes. All right. So here's my HEK flask, and the cells are so dense, you just can't even see cells. They're just like on top of each other, growing so hard. It just looks like a giant mass and mesh of cells. This is way too far. If your cells look like this, it usually means they've grown way too much. You definitely want to passage your cells before they get to this point. But some of you might, your flasks that you've gotten and grown, they might get to this place. That's okay. We're going to figure it out today. We're going to work with it. We're going to make it happen. So generally what I do is I have a waste container. I use just like a bottle, but you can also just as easily use just like a 50 ml tube or something. Like that's no problem. And so what we're gonna do today, ooh, you guys couldn't see me. I use like a bottle, something like this. But you could also just use like a 50 ml tube, something like this. Basically, you just want a place that you can dump out and pipette your waste. So all your media and stuff like that. The first thing we're going to have to do is get rid of the media in the flask. Now, there are proteins in FBS that actually inhibit trypsin. And that's probably like the biggest thing you're going to have to overcome in passaging is just getting rid of all the FBS. Otherwise, it's pretty easy. It's pretty easy. It shouldn't be that difficult. All we're doing is basically moving around media. So I'd highly recommend that whenever you're working and opening up flasks, you wear a mask because you're a contamination vector, you dirty, dirty people. I am also, so I'm not going to, I'm not judging here. Not at all. Not judging. So what I like to do is take my flask with media and just dump out the media. That's going to be the first step. Just dump it all out. There's probably going to be a little bit left. What I do is I'll take like a paper towel or something and just like try to get out the rest of it so that there's not much media left in there at all. Pretty much all gone. And then we're going to take our 1 ml pipette. Now, trypsin, you don't really need to sterilize because it's an enzyme that breaks apart proteins. So usually organisms can't survive well in trypsin. I've never had a contamination issue come from trypsin. So I'm not saying it's not possible, but I've never experienced it. So generally, you don't have to sterilize the trypsin. But you should sterilize your pipette, and you should sterilize everything else. All right, so I got my pipette, got a pipette tip. I'm going to take 1 ml of the trypsin and squirt it in the flask. And just swirl it around, remove any of the excess FBS and media that was in there, and then just dump it out. So it should be totally empty. All right. And then what I'm going to do is I'm going to add 1 more ml in there. And then I'm just going to stick it in the incubator, right? So the trypsin works best at warmer temperatures, so it'll work better in the incubator. So if you just take your flask with the trypsin in it and stick it in the incubator for a couple minutes, the cells should start to detach. And so we're going to wait on that. Now, for the media, you want to warm your media up. So if you just took your media out of the fridge, you want to put it on the heat mat, stick it in the incubator, even microwave for a few seconds, but don't microwave it too long. Microwave it for like, I don't know, 5 or 10 seconds or something like that. Don't microwave it too long though, because you don't want to heat it up too much. I know my hair always gets in my face and I need to get a fucking haircut. Ooh, sorry, I used profanity again. I need to get a haircut. Please, just work with me. But yeah. So what was I talking? Oh, media, warming up your media. So cells generally they like warmer temperatures, right? So if you put cold media on them, it's generally not going to be good. I'm not saying I've never done it. I'm pretty sure I have, but you want to treat your human cells with a lot of respect and love. You want to treat them like they want to be treated. And that means keeping the temperature of the media at a reasonable temperature, near 37 Celsius. So what you want to do is take your media and stick it in the incubator. You can even microwave it for a few seconds. Less than 10, I wouldn't microwave for any more than 10 seconds. If you do, you're probably going to mess up something, so don't. But stick it in the incubator, warm up your media, so then after, when we've trypsinized the cells, everything is ready. It probably takes about, I don't know, I would say like a good, at least if you microwave your media, at least a good 10 minutes after you stick it in the incubator for it to like acclimate. If you just stick it straight from the fridge on the heat mat in an incubator, you're probably talking more about 30 minutes. So give yourself some time, start early, make sure your cells are, make sure your media is warm so your cells are happy. Right on. Any questions so far? Any questions while our trypsin is doing its thing? There's like a little bit of lag. It always bothers me because you're saying what I said like 30 seconds ago. But that's okay. Is Star San a good sanitizer? Sure. I don't think there's really like, I just use ethanol, alcohol. So, it's up to you. Just keep stuff clean, It's not the end of the world. How long in the incubator? 5 minutes? Yeah, generally I start out with like 5 minutes. It's nothing specific. You're just basically trying to keep it in there until the cells fall off. So, 5 minutes is a good time frame, but if you do 10 minutes, it's probably not going to hurt anything. Not with HEK cells, at least. They don't really care that much. So, just put the cells in there until you see them starting to fall off. That's about the only thing you can do. I just poured it out. I just pour out the flask. Do you need to pour it out in any specific way? No. Or dispose of it? No. Generally, I keep a little bottle that has some bleach in it to kill any of the cells or anything like that. But like human cells can't really escape into the environment. You're not worried about mutant human cells going in our wastewater and attacking people. But you never know. So, but yeah, you can just pour it out, wash it down the drain. All right. We can check on our cells. Let's see how our cells are doing. Ooh. Yeah, you can see. Let's see. Shake it up. Shake, shake, shake. You can see how opaque that solution is now. How do I get focus on it? Focus. Focus. All the cells fell off, and they're in the solution. You can see it's like cloudy. We have like a cloudy solution, right? So here's the trypsin and what it looks like. It's clear. And then we got our cells, and it's like cloudy. So yeah, we got our cells. They're broken up. Put back on the mask. Yeah. Put back on our mask so we don't contaminate everything. Wipe everything down again because we touched everything, including my hair and face and all this other stuff. And now we're just gonna break up the cells. So what I do is just pipette them up and down a little bit because they're still going to have clumps, and maybe like spray the bottom of the flask just to clean off any extra cells. We're just going to pipette it up and down to break up the cells. Now we got a lot of cells in this flask. A lot of cells. We don't need that many cells, but that's okay. 2 empty flasks. And we're going to need, you should have gotten syringes, sterile, and filters. So syringes and filters, great, use them. We sterile filtered the media, but there's always going to be something. So the way you put on the filter is it just screws into the bottom of the syringe. So you stick it on the end of the syringe, and then you just turn it, and it should lock in. It should just like lock in after you screw it. And then what I do is I take the media. Well, first let me take out the bottom of the syringe. I just sanitize my pipettes all the time, even if I'm not worried it's contaminated. It's just like a good practice to have. Just spray everything with ethanol all the time. Like it's not going to be bad, and it doesn't take much effort. But yeah, you should, wipe down your pipettes as much as you can. So I just take the media and I just dump it into the end of the syringe, just like that. Just dump some media in the end. Now, we have trypsin with these cells, and we want to neutralize the trypsin. So generally, you want to use about 4 or 5 times as much media as you do cells with trypsin. So what I'm going to do is I'm going to take a flask, and these syringes, they have ml markings on them. I'm going to put in about 3 mLs of the media. So you just push down on the syringe, it'll go through. And then I'm going to put in 3 mLs of the other media. I mean, in the other flask, right? Put it down in there. And then leftover media, you could just syringe it back into your media container if you want. You can throw it out. It doesn't really matter. I wouldn't reuse the syringes and filters because once they get media on them, they can get contaminated, right? Because like everything going out this way is sterile, right? But once you have media on this side, from the air, fungus and other mold and other shit like that. Oh man, I used profanity again. Stuff like that will come up in and start to grow because the media has sugar and stuff in it. How come the filter seize up when they hit an air bubble? I don't know. But generally, if your filter is starting to seize up too much, and it shouldn't be hard to push. So if you're pushing your media through your syringe filter and it feels like it's really hard and a struggle, something's wrong. Change your filter, change out something. Something is wrong. So try to figure it out. Try to contact us if nothing else happens. All right. So now profanity works. I know, but I'm trying to at least make these classes like semi-friendly to everybody who wants to watch them. All ages audience. I'm going for an all ages audience here. Yeah. I'm doing my best. So I cleaned off my pipette again. I'm going to use it again. Now what we're going to do, so depending on how dense your culture was that we split or passaged, if it's really dense, like really confluent, and like the whole flask is covered, I probably use more like, I don't know, 200 microliters of cells or something like that. That's still probably a lot. You could probably go down to like 100 if you want them to take a little while to grow. So I'm going to take 200 microliters. If you've never used a pipette before, you know, we have guides on that, or look up online. They're not as intuitive as you would hope, so don't just give it a shot if you've never used it before. At least try to learn. So I added 200 microliters of cells to one flask, and then I'm going to add 200 microliters of cells to another flask. And then if you want, what you can even do, make sure you write dates on these flasks. If you want, what you can do is you can actually take media and add it back to the trypsin flask. So the trypsin flask, there's still some cells left in here. And if we want to have another extra flask just in case, you know, take a couple ml of media and just add it back, add a couple ml of media to the trypsin flask, and then those cells should reattach and grow. Is there any reason you'd want the cell to grow slowly? Yes. It depends on the timing of your experiments or what you want to do, right? So you might not always want your cells to be ready in like 2 or 3 or 4 days or whatever. You might want your cells to be ready in like 1 week or 2 weeks or something like that. You know, you will use less cells depending on the timing of your experiment and what you're doing. Let me see if I can find, do I have a marker so I can label these? How often do you clean, re-grease the pipette barrel? Like never. You shouldn't have to, generally. It's not a very common thing. You can calibrate your pipette. That you might have to do every once in a while. But generally, the pipette, you know, maybe if you have it for like 4 years or 5 years or longer, you might want to give it like a tune-up. But generally, you're probably okay with not doing it. So I'm going to take my cells. So I have my trypsin cells, and I have my 2 new passage cell flasks. And I'm going to stick these in the incubator. Now we've tripled the population of cells we have, and we have extra flasks just in case anything gets contaminated. So when we go to do experiments starting next week, we have a lot of cells to work with, right? Because we're going to try to kill off some cells. I mean, that's basically what genetic engineering is you're going to, you know, select for a genetic change. And when you select for the genetic change, you're going to kill some cells. So you want some cells to experiment with, some cells to kill, and still have like a flask of cells that's growing as a backup or just as like a culture that you can use whenever you want. So we're going to stick these in here and let these cells grow. All right, so we got the cells. Basically, this splitting and passaging, you're going to do this the same way for almost every cell type. Some cells are a little more sensitive than others, so you might use something else besides trypsin. But generally, every single cell type you'll ever work with, stem cells, you know, primary tissue culture, all these different cells, you're going to do the same exact thing to grow and proliferate your cells for experimentation. You know, even if you're trying to grow, say, like chicken muscle cells to make a chicken nugget, you're going to do the same exact thing. So being able to do this, and as simple as it seems, being able to do this is like one of the foundations of mammalian and human cell culture. Being able to passage and split cells. Because basically all you're constantly trying to do is grow up a lot of cells so you can experiment with them and do your experiment. Grow up a lot of cells so you can experiment with them do your experiment. So yeah, it is interesting when you're growing, human and mammalian cells because they respond differently. You'll like take two flasks with the exact same amount of cells, put them in the exact same incubator, and like one flask will just like die or not grow well, and the other flask will grow just fine. So sometimes it's almost like they each, they have their own like personalities, and that's totally me projecting. Probably, maybe, I don't know, but it's kind of me projecting. Um, so but you do develop this like weird relationship, because you spend time with them, you're cultivating them, you're taking care of them. Um, so I definitely get more attached to like human and mammalian cells than I do to, bacteria or yeast or things like that. Um, Visible mutations, that's hard. What you will see is senescent cells. So if you keep your HEK cell culture alive for a long period of time, um, what you'll start to see is you'll see these like big kind of blob looking things in the middle of your growing cells with like maybe a giant nucleus. Um, and it's just like this big circular looking thing. It's a senescent cell. So it's a cell that just kind of like uh, reached its Hayflick limit or whatever and is just dying. Um, it's no longer going to be able to proliferate because, right, HEK cells are immortalized. So technically they can grow forever, but it's just technically they can't actually grow forever. They still run into mutations and errors and things like that. Uh, so the cells do senesce eventually. The cultures do get overburdened with mutations. It's hard to tell if you see mutations in individual cells because, the cells they look fairly, I don't know, how do you say, like, not every cell looks the same, right? They're very diverse in their appearance. So I think it would be hard visually alone to tell if there was a mutation or something like that. Um, can you use the same media even with dead cells if the media is still pink and viable? That's a good question. People do actually use media. They call it conditioned media, which is like media that cells have grown in. Um, you can use that to uh, grow other cells in. Now, the problem with the media is like just because it's pink doesn't mean it's still good, right? There's a lot of other stuff that can happen in the media, that can go on in the media, that's not easily visual. That's not just visual by the color change of the media, the pH dye. So I wouldn't necessarily use pink as a synonymous with good or viable. Pink, I think, just means that the pH hasn't changed enough for anything to be different. And yeah, it could mean a lot of things. So it's possible. It does work. I would sterile filter it probably before I used it. But personally, I would just rather use fresh media. I've tried it before to use conditioned media, media that cells have grown in before, and it didn't seem to work very well. Um, how long does the media last in the fridge? It depends how sterile you are. Because the media should last indefinitely. Usually the number one reason the media doesn't last indefinitely is because you contaminated it. So yeah, the media can last forever. You can't freeze the media though. When you freeze it, some of the components will precipitate out, and so it's not good to freeze the media. Um, but it should last indefinitely, basically, in a fridge. We got some good questions here today. I like it. Yeah. Why do non-immortalized cells senesce even after passaging? So passaging is just a way to break apart cells, reseed them so that they grow. So passaging isn't necessarily a lot of times people will refer to a cell line as like what passage number it's on. So like this cell line is on passage number 0 or passage number 1 or passage number 5 or something like that. But you have to understand that like the cells are multiplying probably a few times before you even passage them, right? So it's not a great indicator of like age or much in the cells. So why would a non-immortalized cell line senesce? So generally, senescence has to do with the cell's DNA or the cells in general, accumulation of mutations or things like that. Think about your own body, right? Your body, the cells in your body are being replaced all the time, right? They're dying, and then new cells are growing and coming out, right? And the cells that are dying is the senescence, right? So this is happening constantly in our bodies. It's constantly going on. And it's going to happen in cells in culture, right? So the cells are going to accumulate mutations. The cells are going to reach their Hayflick limit, their telomeres, and they're going to die. They're just not going to be able to be viable for much longer. Yeah, so cells, non-immortalized cells, will generally, depending, aren't going to last very long. Immortalized cell lines can last very long, but they still accumulate mutations and stuff like that. So they'll just be messed up the further along they are. So I just pulled my trypsin out of the freezer. Is there any prep? Just let it thaw. There's nothing you really have to do. Put it on your heat pad or stick it in a bowl of warm water or something like that. Just thaw it, and it should be fine. Yeah, try to warm it up to at least room temperature for doing your experiment. All right. Anything else? Any other questions about human cell culture? Proliferation, engineering. Yeah, so next week we're going to start working on how to select for genetically modified cells, and that's using antibiotics. So chemicals that'll kill the cells unless they have certain genes or proteins. Once you know how to do that, then you can genetically modify. Should we keep trypsin in the fridge or freezer? It doesn't really matter too much. Fridge and freezer both work. It depends on how long you store it and what you're going for, but fridge and freezer should both be okay for your trypsin. Like I was saying before, I've never seen contamination or organisms in my trypsin. Like I've never done a cell passage and then had contamination. Because trypsin just chews up protein, and if you leave any cells in it too long, it'll basically just destroy those cells and everything associated with them. So it's generally okay to leave it in the fridge or freezer. Honestly, if I'm being honest, I just leave my trypsin on the lab bench. I don't put it in the fridge or freezer because I've never had any issues with it. But that's just me. So don't do what I do. Do what I say. Especially if you don't have a minimally sterile working environment. Could I recommend reading material that might be good to accompany this class? Not really. This class is about techniques rather than specific scientific data or theories or information. While I'm sure there's like books out there about human cell culture and stuff like that that you could find and read it. To me, a lot of science is just about hands-on experimenting. You understand the principles, you understand what's going on, and you can do it with your hands. That's basically it. There's nothing, I mean, obviously having a general knowledge and understanding of biology and cells and stuff like that is great. But in general, I don't know specifically if there is much things that need to be learned besides what we're teaching in the class. And if you have questions, feel free to ask. Feel free to post the questions in our Facebook group and stuff like that. Yeah. Yeah, there is fungicide that you can use. Amphotericin is a good one. I don't know if we started adding amphotericin to the media. I'll have to ask David. He's the one who makes the media for these classes. He might have added amphotericin to the media. So, that's a good one generally to stop this stuff. But would you be able to point us in a direction on how we might try to culture our own primary non-immortalized cells that we collected? This really depends on the cell type. That's the thing. So you can imagine that there's so many different cell types in your body. And so all the cells have different requirements for what they need to grow. So depending on the cell type, that's what's going to change the requirements or how you grow it. So what I would do specifically, depending on what you're trying to grow, is first search the internet. Find procedures and protocols that people use for that cell type. Like your cheek cells, they don't really proliferate and grow, right? So like you can take your cheek cells, scrape them, put them in a culture, and start and like keep them alive. They'll even attach to the flask, but they don't really proliferate, and they'll die after like 1 week. But it's totally possible, right? That's different as opposed to like fibroblasts, right? So things you get like from like your connective muscle tissue. Most of your, there's a lot of fibroblasts in your body. Now, fibroblasts require certain hormones. Generally, people use FGF and EGF. You can find those and purchase those online, and they use slightly different media conditions. So like in this experiment, we're using 10% serum. So we use fetal bovine serum. Fetal bovine serum actually comes from fetal cows, which is kind of gross. We've been trying to use other serums for this class, but fetal bovine serum right now is the easiest and works the best. So in class, we use 10% fetal bovine serum. In serum, there's like hormones and growth factors and other things that seem required for a lot of cell growth. You use 20% serum with fibroblasts generally. It helps them grow better. So there's all these different things, little things, and depending on the cell type that you'd want to use to do something. Would fibroblasts be one of the easier to start with? What is a good starter tissue? HEK cells are great starter tissue. So if you're just getting into human cell culture and you're working on this, first I would start with HEK cells. Take part in this class or buy HEK cells and learn to culture them. Once you've learned to culture HEK cells, then I would start moving on to other cells. But before that, I wouldn't. I would just focus on HEK cells until I had that down really well. But yeah, fibroblasts are good starting tissue working with primary cell culture, but a lot of tissues are pretty much the same. Like you're not going to have that much more difficulty or difference growing like, say, mesenchymal stem cells or something as you are with fibroblasts. They're going to be pretty similar in difficulty. So it depends on what cell type or what things you want to do, but generally the difficulty is going to be within the same order of magnitude. Yeah, so that's one thing I'm really interested in is teaching people how to grow their own cells. One of the problems is that most of the cells that grow and proliferate, it's hard to get access to them. So like if you want to get fibroblasts, you have to take out a chunk of your body, right? You have to do a biopsy or something like that. And that's we can't run a class where people have to harm themselves. So it's a bit difficult. We're stuck at a place where it's like, how would one harvest these cells if we were to run a class where people grew and immortalized their own cells? Because that would be super cool. I think it'd be really cool. I have some of my own cells growing right now. And so, like, yeah. But right now it's no, blood is not really a good source, right? So blood, you can get what they call peripheral blood mononucleated cells, PBMCs. But generally PBMCs don't proliferate really well. Right? So blood has cells that you can get and attach and culture. So just like your cheek, you can scrape cells from your cheek and you can put them in a flask, and they might even proliferate a little bit, but they'll attach to the flask and they'll be alive, but only for like a week, and then they're dead. So if you don't have a specific experiment planned or something like that, you're not going to be able to keep them alive and do different things with them. They're not going to proliferate well and grow and last for a long period of time. So, you really need specific cell types. And usually those cell types you have to harvest in such a way that you have to harm the human body. Yeah, so it's trying to figure out a way around that and getting over that. Yeah, that's a big one. Any other questions? Any other questions about human cell culture, growing cells? I'm here, at least for the next 10 minutes. Will you be teaching how to make induced pluripotent stem cells out of primary cells? I'm not against it, but again, you run into the problem of like where to get primary cells, right? So in order to make induced pluripotent stem cells, generally what you do is it's just it's just genetic engineering. It's just a transformation, right? You're just putting certain genes inside the cell to cause them to de-differentiate into iPSCs, induced pluripotent stem cells. But you need a source of primary cells. You can get primary cells. You can just buy primary cells online. I mean, I guess I could send people primary cells that just bought online, or maybe my own primary tissue cells. Maybe people be interested in that, but that just doesn't sound very interesting to me. And also the transformation process. It's I've never done it before, but generally people require or use special equipment like electroporators and stuff like that. And so like I don't know what the efficiency of it is yet. It might be a fun class, but it's something that we're still working on in inducing our own cells to become pluripotent stem cells. So it's something we're working on, but like, will it ever be a class? I don't know. I hope so. I hope a lot of these. So yeah. Yeah, genes to make the things that are required. Oh, what is it? It's like there's 4. I think they use the 4 stem cell factors. What is it like? NANOG, KLF4, CMYC, and SOX2. Are those the 4 induced pluripotent stem cell factors? Let's look this up. iPSC genes required. OCT4, KLF, SOX2, and MYC. Did I say that? No, I think I said NANOG. Is NANOG the same thing as OCT4? I don't know. Yeah. All right. So it appears to be that MYC, OCT4, SOX2, and KLF4 are the ones required to make induced pluripotent stem cells. But yeah, generally you just buy these pre-packaged in a plasmid or virus from a number of different companies. All right. Anything else? Anything else? Anything else you wanted to know while I'm here? Wasn't Mick the one that was kind of cancerous? Honestly, I don't know. Yeah. What did I type in to find that? If you're talking about the iPSCs, I just typed in iPSC genes required. Yeah. What genes are required to generate iPSCs? But I mean, they have kits. If like, you can literally buy a kit online that will let you turn your cells into iPSCs. So, you know, have I experimented with liposomes as a way to transfect? Yes. I haven't really seen any difference between like all the different transfection methods. They're all pretty good. So, I mean, it really depends on what you're trying to do, I guess. But, you know, I think it generally doesn't matter too much. Unless you're going for like really, really, really high efficiency and you have a really, really, really complicated transformation DNA or whatever. Otherwise, it's like you transform a flask full of cells and you could probably just add DNA with nothing else and you'll get some transformers. So, yeah, I mean, it really depends on the context. Anything. Otherwise, I'm going to go because I've reached my time limit. Oh, it's not a hard time limit, but you know, once we get to the 1 hour point, I don't want to bore everybody too much. Thanks, Matt. Tell Cass I said hi. I'll tell Daria you said what's up. I hope to be safe in Ukraine. I hope nothing happens, but you know, according to the people I know there, things are okay right now, to where I don't have to worry about war breaking out. while I'm there. I hope. But yeah, next week David Ishii will be teaching the class. So if you're like, what happened? Why did you grow a beard and ponytail? It's not me, it's David. Is there a way to save overheated cells? Ethan, I don't think your cells are overheated. The pictures you sent, they're just dead. Just contact us and we'll send you new cells. Yes, the videos are available after streaming. Generally, if you go to the Odin YouTube channel, youtube.com/the Odin Inc. INC, there should be a playlist for growing and engineering human cells with LAC-Z, which is different from the growing and engineering human cells class. That was our old one. This is the new one that we're doing with LAC-Z. There should be a playlist where you should be able to find all the previous classes. So yeah, the classes should be posted just directly after. I mean, the videos should be posted just directly after the live stream. So you should be able to find them there. So you can rewatch the class. If you have any trouble getting access to the course materials, the protocols, the Facebook group or anything like that, just email us, odin@the-odin.com. We'll help you out. We're here for you. If your if anything happens with your materials or anything, just please contact us. We're here to help you out. We're here to make sure that you're able to successfully accomplish this class. So media gets contaminated, cells die, whatever. Just contact us. We'll send you stuff just for the cost of shipping. Right? So just contact us. We'll make it happen. The software I use is AM Scope software. It's the same exact thing. So yeah, sorry about that. But yeah, anyway, I appreciate everybody for coming. Come back next week, same time, 11 a.m. Central. David will be here to teach you more about selecting cells for genetic modification. Otherwise, I'll be back for the last class where we actually add DNA to do the genetic modification and then look at the results. All right. Anyways, catch y'all soon.